this. All right, so today's a review session. So we're gonna go over the practice exams really quickly, and then we'll again, quickly just to review the important points, and that especially for you guys for preparing the exam. All right, so as we have done, we're gonna just go over the exams, and whenever you have questions, just let me know. Otherwise, for the simple ones, I'm gonna just quickly skip them. So the first one, the right answer is A, and this is nothing but you just simply just memorize the structure or identify the actual like structure for the their specific confirmations. Number two, what's the right answer? C. And if you don't understand, I mean, basically you can see the structure of a cholesterol and can easily tell that there are four rings. Number three, A or D, it's D. And so I guess, I mean, everybody got that the A is correct, where all the polysaccharides have one or more reducing end. What about C? Some polysaccharides may have one reducing end and many non-reducing ends. Is it correct? I've actually explained this during my lecture, where all the polysaccharides contain one reducing end, one. So that is correct. Number four, B, E. All of them, basically. We talked about this, how it sets up the gradient or the basically what's it, the driving force for the making this kind of fluxes. Number five, it is C. And essentially, you're looking for the most non-polar one, right? Which can just go through the lipid bilayer without any kinds of losing much of the energy there. Not losing energy, but at least it doesn't require much of energy to go through that. Number six, it's B. So again, you can go back to all the descriptions about the different kinds of polysaccharides, and you can get an idea, basically. For the rest of them are containing all the 1 to 4 connections. Number seven, and the reason why is because it's seen in liver and muscle in animals, right? So it has to be in glycogen, not the amylopectin or any other things like amylose, basically, which is also the homopolysaccharides. Number eight, C. And it is important, right? Because you need to be able to distinguish what are the chiral centers. And the chiral center numbers are different from aldosis to ketosis. Question. Yeah, yes and no. Basically, it actually says that aldopentose. So aldose, you kind of know the structure where it has aldehyde group on the top, and CH2OH on the bottom, and then the number of C's that you can basically come up with based on this pentose, or the hexose and things like that. So you technically don't need to memorize all the structures where the hydroxyl group is whether on the right side or left side, but at least you can tell how many of the carbons that comprise this. All right, so number 9. I mean, there's nothing to explain other than you guys just identify and distinguish the different sugars. Number 10, B, anomers. And I spent quite a bit of time for explaining what the anomers are, what the epimers are, and things like that, right? So you should be able to distinguish between those two, and of course, should be able to tell that if such structure is anomer or the epimer to other structure and things like that. Number 11, cellulose, C, basically. Again, this is just a fact, right? Number 12, B, Again, we talked about this during the lecture, so it's all the properties of the polysaccharides, right? As well as some monosaccharides too that you need to basically aware. Number 13, C. Again, this is the only polysaccharides that are used for bacterial cell walls. Number 14, it's E, and N-linked because it has to contain amino group, right? And asparagine does that, as opposed to all others don't. Number 15, D. I think I heard D. So same thing applies here, right? All linked for the hydroxyl group. So any nucleotide amino acids that contain the hydroxyl group will be potentially make the all link linkages. But we have threonine and serine that are mostly used for that linkage. Number 16, what is it? D? How come? There's a question. Good question. Why not other things other than D? Can anyone explain? Hold on, was it about number 15? Oh, I'm sorry. I thought it was number 16. Sorry about that. So what was the answer for that? That is correct. So again, only serine and threonine, which have the pretty prominent hydroxyl group presenting right towards the outside of the side chain, right? So it can be easily linked there. Tyrosine does have that, but it's much less active because of the phenyl ring. Does that answer to your question? Sorry, number 16. Going back to number 16, someone says, some of you guys actually said D is the correct answer. Why? Why not A, B, or even C, or even D, or even E? Yes. Negative phosphate group makes the head group polar, right? So when you have a phosphoryl group, then you're going to have a polar head. Does it make sense? That applies to glycerophospholipids, of course, because it contains a phosphoryl group. What about sphingomyelins? Has anyone actually looked at the structure? That is you're completely right about that, but it actually doesn't specify the cell type here. It's just saying that the major lipid components of the cell membrane, so it can be applied to the nerve cells or other cells. Amphipathic. Sphingomyelin again contains a phosphoryl group. It's obviously polar. It has a negative charge, right? PO4, PO4 minus. So it does actually a lot, like 20% or so. It's a lot. So the right answer here is all of them, A to D. I've actually written that in a way that just D is the correct, but the just have a little fun there. Wanted to make sure you guys are thinking in the right way. But all of them are amphipathic, meaning that they have at least a partial charge on the certain region, certain group of their molecule, basically. Sterols have a hydroxyl group attached there, making a polar head. And it's a major component of cell membrane It has the like 30 or 40% in certain cell types. So that's a lot. Sphingomyelin, of course, has the phosphorus group. Gangliosides has sugars. Sugars have lots of hydroxyls. All of them have capacity to lose proton and become polar. So all of them basically, except for triacylglycerol, which is completely non-polar. So this is the right answer. I could have asked in the opposite way, right? What are the one that does not belong to this class, something like that. Number 17, D. And does anyone need explanation for this? Because everything is on the slides. I just simply use the old words, basically wordings on the slides, and then change it a little bit, like the here fewer than 14, but it's around 16 to 18, right? And fatty acid is synthesized from carbohydrates. It's incorrect because it uses a different pathway that we didn't even discuss, right? Which is acetyl-CoA or coenzyme A. And the conjugated double bonds are most common fatty acid types, which is not true. Number 18, D, right? And again, you just have the property, understand the property of unsaturated fatty acids and make a comparison with the saturated ones. Number 19, A, it's pretty simple, right? Why not B? Adipocytes are specialized cells for short-term energy storage. What is it? It is a long-term. Short-term would be glycogen, which happens in liver, as opposed to adipocytes are on the connective tissues, right? Storing it for long-term in the form of fat, basically. Question, was there a question? Or? Number 20? That's correct. Number 21? D. Again, you just have to understand, just have to know all the difference between these different lipids, basically. Number 22. Yeah, I hope that was a relatively simple, straightforward kind of question here. Any questions here? Number 23. A. Why not B? Non-mediated transport sounds like something kind of passive kind of thing. What about facilitated diffusion? Precisely. So there has to be a channel or something that goes through the basically all the stuff, but in a passive way, meaning that the ATP hydrolysis isn't involved there. 24. B. I think I heard B there. Question. Good question. So what? Absolutely. So the question was that why is the D is not a correct answer, because when you look at the neurons, nervous system, basically everything happens based on the voltage changes, right? Can anyone answer to this question? No one? Or anyone? Make a wild guess. Sometimes it is charged, but mostly they're not charged. That's for sure. Actually, that is a partially correct answer. So if it is not charged, probably not induce the voltage. That could be an answer, but there's a more direct answer to that. What about the voltage-gated channel? So how does a neurotransmitter work? Yes. So that is correct. When you think about neurotransmitters, it does not bind to membranes or the any kinds of carriers or channels and things like that. It binds to synapses, binds to the specific protein on the specific locus, which is different from the membrane per se. So it does not work in that way as opposed to, it has to just bind to the specific like receptor. So it's a ligand receptor kind of binding, as opposed to voltage gated is simply just opening up the channel based on the differences of the, electrostatic potential there. Sorry. Yeah, you first. Well, that's a good point. I mean, can anyone actually try to answer to this question? Why not E? I mean, everything has to be affected by some kind of stimuli, right? Whatever that is. Extracellular stimulus. That's the catch. But can anyone try to? Yes, so some of them can be happen intracellularly too, and exclusively intracellularly. Voltage gated, I mean, it just happens, I would say, intracellularly because there's nothing coming outside. It's just the some kind of signaling pathway triggers the internally that opens up the K like leak channels and things like that. So it's more like as opposed to ligand, it has to be always come from outside all the time. Question back there. Okay, good. Number 25. Oh, sorry. Question. I'm sorry, I didn't catch the middle part that you're saying. That is absolutely true too. So what she was saying was basically something happens in the opposite way, by the receptor ligand interactions. So is that possible? Was that the question? Yes, the answer is yes. I'm just to try to find the examples, actual examples. Yeah, I don't know. I can't think of anything at the moment, but there are cases where they work as a sort of a competitor of the other active ligands. So that's one example. I mean, you can take it as a drugs too. And also, it is always open, but depending on the interaction with the some other protein like a ligand, it actually makes into experience the conformation or change when it becomes to the closed form. So that's definitely possible too. And there are many cases, I mean, that I can't think of right now. Sorry, no question. It can actually. When you go back to my slides, it actually can come from the intracellular too. So it can be both ways, but in the most cases, it happens from the extracellular outside of the cell. But that is true. I mean, there's no such thing as never, in biology, right? So it happens both ways. Okay, number 25. C, and the region. Precisely. So when you look at it, I mean, it usually happens in cis, right? But now you can see that it's in trans, meaning that it's actually localized in a different, the other side, which is really rare, really, really rare. So that's why. Number 26. What is it? Two different molecules in the different directions. Antiport or antiport. And again, I mean, the answer antiport isn't that important. The important thing is that whether you understand other things like symport, uniport, gap junctions. There's no such thing as equilibrium channel, but there's a leak channel, right? Makes the equilibrium. 27. D, I think I heard D. So again, as long as you understand the symport, uniport, and the active transport or the passive transport, you should be able to answer this question. 28. C. And I don't think any other things that is confusing. Again, I just simply used all the wordings that I used during my lecture, and then just to change a few words there, like only in animal cells, something like that, or the soluble instead of insoluble in aqueous solution. Number 29, C, right? Because it's in a leaflet. Again, I just changed a little bit, just one word there. It's going to be a little bit more obvious when you're doing actual exam, but what I want you to do here is not about just picking the right answer and move on. I want you to actually, again, I mean, obviously you have to just see all the linked proteins there and go back and review all those, and also looking at all the details here to make sure you're ready for the exam. Number 30. C. Again, little facts, right? I'm not gonna do this kind of thing, like changing just a little one bit there. But again, all these facts are important. So, all right. So, any questions so far for the exam or anything? So I already talked to you about the grading scale and the weight of the midterms and finals and things like that. So if anything's unclear, just let me know. And also, this exam will include the 3 bonus questions, so you can get up to 110 points again. So good luck with that. So from now on, we're gonna move on to the review. Let me just kind of. All right. So we first started with the carbohydrates, especially monosaccharides, and how you can write it in the Fisher projection, what it means, how you can tell the L forms and D forms. And the numbering too is important, right, for the aldoses and ketoses here. And also we talked about enantiomers and other stereoisomers too. So you should be able to tell if that is enantiomers or other type of like isomers, basically. And we also talked about epimers as well as anomers. I'm not sure if we can actually find it here. What epimers mean and how those 2 epimers can relate it to each other. And anomers too. I'm not sure if I actually included here, but anomers are really important, especially when it's forming the cyclic structure. The anomer, you should be able to identify which carbon is an anomeric carbon. We covered the structures of aldoses as well as ketoses here. Again, memorizing the structure is important. The more important thing is that you can relate the one molecule to the other in terms of their structures, like anomers or not the anomers but epimers or the enantiomers and things like that. We talked about the cyclic form and the alcohol and aldehyde within. So it's an intermolecular interaction to form the cyclic structure, and it makes this hemoacetal or the hemiketal or hemiketal functional group here or structure here. And you can tell whether this is alpha or beta based on their anomeric structure. And of course, we talked about the anomers and how many anomeric centers you can have. as well as how it can actually forms the alpha or beta forms, and the transition or the basically it can go back and forth, right? Alpha form to beta form. It doesn't just simply flip it, but it has to go back to linear form and have to get the different anomeric structure. Question. The answer is yes. So aldehyde should be the CHO should be always on the top, and it'll be numbered. The numbering basically starts from there. So and when you talk about the it's D or L form, is that your question? Got you. So this, right? In this case, I mean, it's a little bit complicated. It's really hard, actually. It does not have any kind of distinguishment between the up and bottom and things like that. So you can actually, honestly, I don't recall anymore because I've never used the Fisher projection. I've never seen anyone use Fisher projection for indicating the cyclic form. So I don't know, but they promised in a way that the left-hand side is always on the bottom and stuff like that, but it's not really working too well. So instead of using that, we use this Howard projection, where it is more intuitive, right? And I don't actually use Fisher projection for showing the cyclic form, cyclic structure, so don't worry about it. Question. I said why? Okay. So what I just said by answering the question, what I said was that the question was whether how you can tell which functional group is on projecting to the top or the bottom, looking at the Fisher projection of the cyclic form, which is looking like this, right? And this is different from Howard projection that you can see here, in which case you can see clearly where the functional groups are projecting, right? In here, there's no way, basically. So I think I remember the day sort of use the convention where left-hand side is always on the bottom and the right side is on the top and things like that. But again, I've I've never seen anyone uses this Fisher projection for showing the structure of the cyclic, sugars or the rings. You focus on cyclic ones. Um, yeah, I mean that's important because that's the structure of sugar, right? Simple sugars. So you should focus on that. But rather than using this Fisher projection, you can use Howard projection. Oh, got you. So um, D or L form is based on the chiral center, right? So it's gotta be in the linear form like this. So to be able to tell that this is whether D form or L form, you have to take into account of this linear form, especially for carbon 5, whether hydroxyl group is on your right side or left hand side. If it's on right side, then it's D form. Left side, then L form. As opposed to for cyclic form, now you talk about anomers, whether alpha or beta, and depends on the orientation of this hydroxyl group that is attached to the anomeric center or anomeric carbon and the extracyclic carbon. It, mhm. So that's gonna be this carbon, right? Here. Mhm. Um, no, OH is always here, so you're not gonna have D or L form there, but it's based on its linear form. So when you make it capture the linear structure, then you can tell whether this is D or L form, and that's the origin of this cyclic form, right? Which can tell the alpha or beta form. So you're gonna have both like beta D or beta L or something like that. But this D not necessarily tells the difference on the cyclic form because it's going to have the same structure here as CH and O, like that. Does it make sense? All right, so we talked about this hemicidal structure too. And then anomers, of course. And we talked about the properties of sugars, especially it makes this linkage using the glycosidic bond. And this is different from the N-glycosidic bonds that is used for this making nucleotide structure. And we also talked about the disaccharides, their structures, their nomenclatures, and the reducing end and non-reducing end, the difference between those two, right? And then we moved on to the polysaccharides. And I'm not going to go over all their properties and structures and things like that, but you should know the cellulose starch, which is consistent of amylose and amylopectin, and glycogen, their differences, their similarities, and their structures, their linkages, where they are used, and things like that. And we talked about the glycoproteins, especially, talking about these linkages where it uses the hydroxyl group or the amino group between the amino acids, side chains, and the sugars. And also what kind of, amino acids are involved there, here. And peptidoglycans that are used for cell walls for bacteria. And we also talked about all different kinds of lipids, fatty acids, triacylglycerols, phospholipids, which are really important for forming the biological membrane, sphingolipids, waxes, and so on. And steroids are important too, forming the biomembrane. And talked about the different kinds of saturation, right? Saturated, unsaturated lipids, and how it causes differences in the properties. And again, we talked about the lipids, especially, talking quite a bit about this triacylglycerols, glycerophospholipids, and sphingolipids. And this glycerophospholipids is important because it contains a phosphoryl group, which makes the head really polar, right? And that is an important property for getting into the biomembrane. So again, triacylglycerols, we talked about it. Glycerophospholipids too, as well as sphingolipids, their structures and where they are used. And glycosphingolipids too, which are cerebrosides and gangliosides, their structures, what they're attached to as a head group. And steroids, their structure. And we also talked about micelles, lipid bilayers, as well as liposomes, how they can be formed, right? And what kind of lipids are involved for forming those structures. For example, for micelles, it should have a single tail, to make this spherical form, spherical structure, as opposed to lipid bilayers tend to have, two tails. So it's a kind of structural thing where it stabilizes either like making spherical shape or the flat shape. And the liposomes basically contain the little vesicles in it, so it can carry some like soluble proteins or other minerals to other places. And we talked about the fluid-like state of a biological membrane, where this membrane behaves something like the sometimes like liquid, sometimes like more like solid. And talked about the phase transition, how it can happen around the melting temperature, and how melting temperature is affected, and the addition of a cholesterol, how it affects this fluid-like state of the biomembrane. And we also talked about the fluid mosaic model, where all the proteins are stuck there, but kind of floating everywhere in the Brownian motion. And then we moved on to the proteins that are integrated or bound to membrane. So we talked about integral proteins as well as peripheral proteins, lipid anchor proteins, like or the glycoproteins. So these are all about those things. And then we talked about the different type of lipid anchorages. Can use these four strategies basically, using specifically myristic acid or palmitic acid as an anchor. And we also talked about all different kinds of transport, like active transport where it uses ATP as energy source, or the passive transport and passive mediated transport too, which does not use ATP. And even for those transport proteins, and there are different types, right? Like the carrier proteins and the channel proteins, and they have differences there too. And whatever I'm saying here is actually important. So even if I don't explain, fully about these are important topics. And we talked about the passive transport, active transport, what the differences are there, and what kind of systems, what kind of channel proteins are using those transport. And the driving force here too. And we went through each of them, the carriers. There are subtypes like symporters and antiporters. And also coupled the carriers, how it works. Is that against the concentration gradient, or is it following the concentration gradient? That's important, right? Because that determines the whether it requires energy input or not. And we spent quite a bit of time to discuss this sodium potassium pump. So you should understand how it works. And ion channels too, which is more like a passive, right? It does not use the ATP, but it's important for biological functions like the communication between cells, like porins, or also for the equilibrium of the electrostatic potentials to get this action potential in the nerve cells. And potassium leak channels too, we talked about it, especially focusing on this selectivity filter or ion filter. So you should understand how it works too. And that's it. So that's it. So when you have any other questions, just you can come to the office hour today or just email me anytime. And good luck with your exam. So I wanted to ask, what do you think?